Okay, state losses are again, as far as I'm concerned.
Thank God for conversation.
I know, serendipitous, really. I'm so happy to be here. And it's been a couple months and really fun to be part of the cohort with Laura and Devin. Honestly, like having the three of us together to sort of figure things out has made also a big difference and makes it less lonely, because being new assistant professor can be a bit lonely, to be honest. So yeah, it's just nice to have all of us here together. And yeah, I'm happy to have this chance to talk about my favorite topic, which is soil organic matter. And so my goal today is sort of to give a primer on soil organic matter. So I know many of you probably appreciate soils. Maybe the questions you're answering are adjacent to soils in some way, with water or air quality. But today I'm hoping you can leave with some, more facts, more terminology, an appreciation of, greater appreciation of soil organic matter. And so I'll be talking specifically about the role of organic matter in supporting productive and climate resilient ecosystems. And I'll be sprinkling throughout some results from my work. But honestly, the majority of this is almost like, a mini lecture. And then I've, at the end, highlighted some of my research. One experiment in particular that I think showcases nicely sort of the types of experiments I like to run, the tools I use, to give you maybe some ideas of ways we can collaborate moving forward. All right. Because I messed with Zoom, to click here. Okay, and so I want to start with this picture that just to kind of allow us to, make sure we're imagining soil systems at least in a similar way, because I think about soils and their interaction with, organisms, the air above it, the water that's in it. There's lots of interacting parts to it. And this captures many of the components of ecosystems that I think about, not everything, but we have a tractor up top representing agricultural management and the disturbance that creates in soil. You have different types of vegetation, the leaves from the different types of trees. You have the grass as well. Some of them are brown leaves, some of them are green, sort of more tender, all different qualities of plant tissues. You have the above ground and below ground biomass of the plants that I think about as well, the sort of relative contribution of either of those. And then at the fine scale, I think a lot about minerals. And by mineral, I mean inorganic solids like sand, silt, and clay. When you think mineral, you might think of mineral nutrition or mineral nutrients, which is still a fine way to use that word, but I just want to be clear on the terminology. I'm not referring to mineral nutrients, but I'm talking about solids, these really small particles, inorganic solids. And then I also am thinking a lot about the microbes, the bacteria and fungi, the pore space that's created in soil, how they're interacting, the food web, the biology in soil, everything from the fauna, the earthworms, and the mites that you can actually see with your eye, down to, again, the bacteria and fungi. So hopefully we can start picturing this together. And I'll start off with some of the functions that I'm sure many of you are familiar with. And this doesn't cover it all, but some of the major ones is that soil is supplying water and raw materials for society. It's a physical foundation for structures. It's solid earth that we can build upon. Well, usually solid is where we choose to build upon. It's providing nutrients and growing media for food, fiber, and fuel. It's filtering chemical and biological contaminants. It's a major reservoir for microbial diversity. In terms of public health, it's a source of important antibiotics, many, that we still have not discovered. There's also a lot of societal cultural heritage associated with soils. We can also start to think specifically about soil organic matter and the different qualities or functions associated with it, or properties that we assign to it, and how that then supports different ecosystem functions. I like this figure because it sort of categorizes first these measurable qualities of soil as the supporting processes. So it's wide ranging. We have soil structure, including water retention, aeration, aggregation. Soil organic matter is directly related to those properties. The soil biology is directly related to the plant health, the availability of nutrients, carbon storage in soil. We also have the elemental cycles, the biogeochemical cycling of carbon, nitrogen, sulfur, and so on. That is all tied to soil organic organic matter. And then we can go to a higher level of ecosystem function or ecosystem services, it's sometimes called, that soil organic matter provides or supports. And the ones I'll point out are climate regulation that I'll get into, and primary productivity in both natural and managed systems. So a lot of, many different functions and a lot of importance that soil organic matter provides to ecosystem health. And we need to define soil organic matter as well, because you might all come in here with different sort of ideas or definitions of soil organic matter. So I'll generally define it as plant microbial and animal tissues at varying stages of decomposition. And when we're thinking about the soil volume, if you take just, certain given volume of soil, really less than 5%, definitely less than 10% is making up what we call soil organic matter. The majority is the air space in soil, the water that's filling that air space, the mineral inorganic solids, sand, silt, and clay. But organic matter makes up a really small sliver. It includes the living organisms. You might think it's not that important for that reason, but it really has this important sort of outside influence on overall ecosystem function. And so let's get into the definition a bit more. So that was a general definition in the previous slide. This is more detailed, where we're talking about this wide spectrum that spans everything from the carbon that's fixed by plants. When plants are using energy from the sun, fix CO2, create their plant tissues that consist largely of carbon. That enters soil and becomes broken down. And we have everything from that fixed carbon in plants all the way to the CO2 that's eventually respired. Everything in between is soil organic matter. And it's this really, really wide spectrum of chemical and physical complexity, where you have everything from the leaky residues that are just breaking down, that are like on your lawn right now, that are becoming incorporated, to the molecules, individual amino acids, say that a microbe, when it dies, it might excrete different metabolites. Those are organic matter as well. And so we can think about across soil organic matter, these different components, where I've already mentioned that it's composed of plant residues, all the different organisms, and biomolecules, but to think sort of proportionally what makes up soil organic matter itself, less than 5 to 10% of soil organic matter is the actual soil organisms, the living organisms. Less than 20% of that organic matter is the plant debris that's starting to break down. Some might refer to it as particulate organic matter. And then the stuff that's remaining, the bulk of soil organic matter, is what's remaining after microbes have done their sort of decomposing and work on the organic material. It's microbes will consume all the more available carbon and nutrients, and then what's remaining and the microbial byproducts are really the bulk of soil organic matter. And also, when we're talking about soil organic matter, we're often talking about carbon, because soil organic matter is mostly carbon. It's around 50% carbon, with the remaining largely oxygen, hydrogen, and then the smaller portion there, all the other elements, especially nitrogen, sulfur, phosphorus, which, even though it's a little slice there, those are enormously important for sort of the biogeochemistry of systems. But so often soil organic matter and carbon are discussed interchangeably for that reason. And so this is one of the stats or facts that I'm hoping everyone can leave today with in their pocket if you don't know this already. Just that when we think about all terrestrial land on Earth, everything from the tops of the trees down to the bedrock around the world, and all of the carbon that's within it, you might think, the biomass you see above ground is where so much carbon is stored, like in tropical systems with all that productivity. But when we think about all of the terrestrial system, 80% of the carbon is stored in the soil. Okay, so it's this massive reservoir. Another important fact is that when we look at the carbon stored in atmospheric CO2, as atmospheric CO2 versus the carbon stored in soil, in petagram soil is storing over 3 times as much carbon as what's stored in the atmosphere. So just a much larger pool of carbon, 3 times or greater than the carbon that's in the atmosphere, which is in terms of sort of climate change mitigation, really important because this very large pool of carbon, even a very small change in the rate of decomposition will have a really large impact on that number in the atmospheric carbon pool. So, you know, a lot of work on soil organic matter is trying to understand that balance between carbon storage and release of CO2. And so just to simplify it now a bit, soil organic matter is then a really important source of CO2 when it's being decomposed by microbes. You're going to have a lot of CO2 coming out. When that organic matter is decomposed by microbes, it's also releasing nutrients, especially nitrogen, the nutrient plants need the most of out of all other nutrients. And so a lot of, you know, organic matter management is really targeted towards, you know, building this soil organic matter pool so we can minimize and capture more of the CO2 that's in the atmosphere. By say, if we increase sort of the photosynthetic capacity of plants, for example, that's going to increase the amount of carbon that's going into soil. And I'll get into this a bit more later as well. But the main point I'm trying to make here is that organic matter is both the source of CO2 and nutrients, but it's also a really important reservoir for carbon and nutrients. Okay, so we both need sort of the storage of organic matter, but also its decomposition to occur to support plant life. And so we're always thinking about this balance between the accumulation of soil organic matter, especially when we're thinking of sequestering carbon, but we also need to promote its decomposition, right? It's kind of these opposing sort of goals in soil organic matter management. It's this ongoing sort of debate in at least the soils community, how to sort of balance those two goals. And so I like this figure here because it shows we're looking at carbon storage in above ground biomass versus carbon storage in soil across different ecosystems around the world, with the size of the tree or the size of the bar corresponding to the amount of carbon in each. What I like about this is it's showing that it's not just about plant biomass quantity. So you might think tropical systems being really productive above ground, they might store more carbon below ground, but that is not the case. We have this tiny little cool temperate tree storing the most amount of carbon, and in this, you know, boreal systems storing an even greater amount of carbon. And so one of the things I want also you all to take home after this is understanding sort of the many factors that contribute to carbon storage. I think a lot of people have a misconception that it's purely about, you know, more plant litter, more biomass means more carbon, but that is not the case. There are all these other factors in soil that influence how much carbon is in there, stored at a given time. And I'll get into those factors. But I wanted to show, is there another map? That's what I wanted. Okay, so I like this map because it shows that information in a slightly different way, the amount of organic carbon storage around the world in different systems. And what we're seeing is, well, different ecosystems around the world that differ in their climate, the soil type, the biomass quality and quantity. There's just some patterns that emerge. And you can look at the range of organic carbon values that are associated with different soil orders. Has anyone heard of soil orders or seen these words before, like mollisol or, yeah, so same way you have taxonomy and classification with, you know, organisms and biology, you have the same thing with soils, where there's a USDA or there's another separate system. Some countries have their own systems. It gets a little messy, but there's different ways of classifying soils based on their visual properties, some measurable properties, and so we can start to sort of speak the same language when we're comparing soils. And so the highest level in that hierarchy is order. There are 12 soil orders in the world. And these are just a few of them, but I'll pull out just a few as an example. Aridosols are in deserts, arid, semi-arid systems. They have fairly low carbon, well, very low carbon values. You can think of, you know, in the desert, there's less water, less plant life, you know, it kind of makes sense why there's less carbon there. Mollisols are the Midwestern prairie soils, are productive, you know, corn, wheat producing soils. And those have very high, in terms of organic carbon percentage, that is high, especially for agricultural soils, and that productivity of these soils is associated with that high organic matter amount. And so these might look like really small numbers, like 0.1, 1, 2%, what you might see in your soils here, right? I don't know what percentages you see in the farms that you visit.
Somewhere between 0.8 and 3% in general.
Yeah, which might seem like a really narrow range, but there's so much that can change from, say, 0.8 to 1 or 2 or 3 in terms of the function and health of soils. And so I wanted to bring this back because I wanted to focus specifically on this primary productivity box, and just notice how many different arrows go towards it. So it's a little hard sometimes to disentangle, what is it about soil organic matter that's supporting productivity? Because on the one hand, it's helping aerate the soil, it's helping retain water. That might help indirectly allow the plants to access more water and grow better, right? But also organic matter is a source of nutrients. So is it just supplying the nutrients that plants need? There's a lot of different pieces and bounding factors, so it gets a little bit tricky, but we do know in general that organic matter in many systems can help increase the productivity and health and climate resilience of that system. But this is an ongoing debate, because a lot of those, as you can imagine, I think I mentioned, some of these benefits that soil organic matter provides are somewhat indirect, and finding that direct causal link between organic matter and productivity is this ongoing, very hot debate in at least my soil science world, where in general, people do find that when you are at very low organic matter, organic carbon concentrations, whether you're thinking about maize or rice, what we're seeing here, and this is some random forest models. I don't fully understand them, but they're predicting yield using a lot of things. But anyway, this is shown in experimental, field-based data as well, that you see this increase in crop yield with increase in organic carbon, and there's often this threshold where any subsequent increase in carbon is not giving any benefit to yield. And so there's some areas like in Minnesota, and parts of the Midwest where they have really high carbon concentrations already. They're already at this threshold and they're not seeing any additional benefit to yield. So there are folks questioning, should we be focusing on carbon? Should it be other aspects of the system? But I pulled out this quote here because I think it relates to maybe questions around, food security and public health, and maybe to what folks, in our department might be thinking about, and I'll just read it, but although crop yields under the technical potential increase only by a global average of 41 kilograms per hectare, within countries that have a weak or moderate food security, the mean impact is higher. Notably, yield improvements derived from soil organic carbon, once other constraints have been addressed, vary from 26% to 86% of current food deficits in food insecure countries. So this is saying, specifically in food insecure countries or those with weaker moderate food security, there's this even greater benefit to increasing soil organic carbon. With the technical potential referring to if we were to use just current management practices and apply them maybe more efficiently or in a targeted way, you can still increase yield or productivity. But then this unconstrained potential is considering crop breeding and all these different biological microbial stimulants people are coming up with, and these advances that allow for it, potentially this really large increase in benefit to yield. But in general, we do see there's both indirect and likely some direct benefit to productivity. There's also this very important role that organic matter plays in climate change mitigation. So I've touched on this a bit, that soil organic matter is this massive reservoir of carbon. And so it's this sink, people are calling it as well, where we want to add more carbon to that sink and keep it in there. And if we add more carbon in organic matter, there's maybe less of it in the atmosphere, so we can mitigate some of that greenhouse gas emission and concentrations that contribute to climate change. So we could, have this baseline with, the fallow field that's just pumping out a lot of CO2, or we can have some intercropping or more diverse systems or no-till that help mitigate, reduce carbon loss, or even more aggressive, maybe regenerative ag management strategies that help lead to a net carbon uptake in systems. I will say though that this is also a really active area of research and debate, because some are now questioning whether agricultural soils can really offset emissions in any meaningful way compared to other more like, you know, carbon capture technologies or things that people are coming up with otherwise. But like you saw in that ecosystem services or function figure, increasing carbon is not just about this, right? You have all these other benefits that go along with it, with water quality and air quality, or water quality and the biodiversity and the nutrient supply. So I sometimes fear that we're oversimplifying organic matter into this sort of, you know, carbon climate change solution conversation. But there is a way, I think, to reframe it where it's not just about capture more carbon, more better, but also that organic matter is supporting these climate resilient systems as well through, for example, this is one example where, you know, certain soil types or management systems may influence the physical resilience of soils and influence the water infiltration capacity of soils. So, you know, we're seeing much bigger extreme rain events in certain parts of the world. Here in Oklahoma, where I moved from, but you know, soil with poor physical structure that's maybe cemented on the surface because it doesn't have the organic matter to hold these aggregates in place, water will just accumulate, run off, take soil with it, pollutants with it. Water is not moving through. You need that water to be stored for plants to grow and so forth. So that aspect of climate resilience I'm really interested in, especially here. In thinking about how organic matter can help mediate or buffer sort of the effects of, for instance, drought on crop yields. So it's not just about heavy rain events absorbing those. It's also during long periods of drought, which we see more and more of, and that soil retain more moisture for longer, retain biological activity, and maintain it. Okay, then there's a little video. Let's see if it works. Because before I play it, you know, increasing organic matter in general is good, and litter is the sort of main precursor to organic matter, right? It needs to be broken down, have different fates in the soil, but we need litter going into soil to become organic matter. But a very large portion of it is burned off, is respired as CO2. Okay, so what keeps soil carbon in the ground? Why isn't it just turn it in, turn it out? That's what I want to talk about a bit, and you know, what is leading to the net formation over time. And actually, I probably should have played this because it's just a video showing the way, you know, fauna churn soil. There's a lot of vertical movement and fragmentation of leaves that's happening, and over time, these pieces become smaller and incorporated into what you might think of as the actual, you know, soil environment that contains soil organic matter. This is also my argument for don't rake your leaves if you're able to leave them on your lawn and mow them into smaller pieces. You'll be surprised how quickly it'll decompose. I think a lot of people think it'll kill your lawn or invite pests, but it's actually quite beneficial to leave it. It depends on the tree type and the litter quality. I could talk to you about it and give some advice, but this is also my plug for leave the leaves. Leave the leaves, don't rake them. Okay, so what's keeping carbon in the ground? There's a few mechanisms I'm going to cover that I think a lot about in my work that I think translate and transfer over to like pollutant fate and transport to all sorts of problems that many of you are thinking about. One thing I think about a lot is clay-sized minerals, these reactive clay-sized minerals that have a lot of surface area associated with them, a lot of charge associated with it, that provide all these different binding sites for organic molecules that are held with differing strengths and different reversibility. Some of them recycling quickly, some of them they're held with covalent bonds for centuries to millennia, like very strong bonds. But we know that clay composition, types of clays, and their abundance in soils will influence how much carbon organic matter is stored in a soil. And just briefly, why clay minerals are known for this reactivity. It has to do with their very, very small particle size, their high surface area. So they have this high surface area to volume ratio that just has all, so many different binding sites that are made available with, soil, water, and the solutes are passing by. And on top of that, that surface often has charge associated with it, often a negative charge in soils around here, more positive charge in like tropical soils or more weathered soils. Either way, you have organic matter with all its many functional groups, microbial metabolites with all their different charged functional groups that then interact with mineral particles through these mineral organic associations that I obsess over. And then so the mineral associations, because they're so strong, I mentioned some of the covalent ones that can persist for centuries, millennia, that helps protect and keep carbon in soil, right? Once carbon is associated with a clay mineral in that way, it is stable, persistent, we sometimes call it. So that's one mechanism. Then we have physical aggregation. So this is more of a physical protection. So like an occlusion where you have, when you pick up soil, a healthier soil, it has some crumbs associated, right? It's not like a cement fluff. There's like this structure, granular. Those little crumbs are aggregates, and they differ in size in different soils, but they are really important in physically, like in trapping and protecting organic matter. When you look at oxygen concentrations in an aggregate, it often gets into anoxic. No oxygen in the center, which slows decomposition. You often have higher carbon concentrations as a result at the core of these aggregates. And so this is more, keeping the decomposer away from the substrate it wants to decompose, or creating sort of chemical environmental conditions that slow the activity of the microbe itself. So this aggregation is really important. I will mention it's at a different spatial scale. So up there, 1 to 10 microns is more of the mineral organic associations, individual clay particles and microbial cells and individual molecules. But then as you move up into aggregate size fractions, these are up to 10 millimeter visible particles. Okay, so you can maybe start to visualize it. The other, and I guess final mechanism of stabilization I want to highlight is chemical recalcitrance. So these are chemical qualities that slow or prevent decomposition, leading to what many call the selective preservation of compounds in soil. So straw residue, if you put it on your soil in your garden, it's going to decompose slower than clover or grass clippings, right? You might see that in your lawn when you mow and you use the grass clippings, they disappear within a few days, and then brown leaves from an oak tree are taking much longer to break down, and that has to do with chemical recalcitrance, like tougher to break down. You have, at the molecular level, different functional groups and molecular conformations that simply prevent or slow down decomposition. It's why, a leaf, you might see this, has anyone seen this in the woods sometimes, the veins and the tougher materials remaining. And this idea of selective preservation really informs the theory and methods for isolating humus and humic substances. So who here has heard that word, humus or humic substances, and maybe equated soil organic matter with humus, potentially, depending on, yeah. So it's a very common, still used concept and method, but there's actually this whole massive paradigm shift in my world, in my soil science community, of like moving beyond this and actually moving away from this because of the way that it's extracted. So you use a harsh acid or base to extract different types of humic substances based on its solubility in this harsh acid or base. And there's a lot we've learned about soils and their chemistry based on the different amounts of these humic compounds. And it's a good model substance if you're doing other studies, trying to understand sort of natural organic matter and how it affects, other things you're thinking about. There's a lot of different applications. So the issue is that what we're extracting is not representing anything that we're able to measure in soil. It's this very, very high or low pH substance that is creating a substance that is an artifact of the method itself and is not, with new methods, actually measurable or detectable in soil. So there's sort of, this opposition, I'd say, growing towards this that I I need a whole other seminar to talk about, but I'll just share that. So soil organic matter, as you can see, is this wide spectrum, very heterogeneous. You have different sort of mechanisms that are stabilizing organic matter. And so we often use fractionation to start to separate that complexity into more distinct pools. And there are lots of different ways of doing it. Unfortunately, I think, 20 plus different ways that people are separating organic matter, chemical, physical, and so forth. But they all, to some extent, are reflecting these different mechanisms of stabilization, like whether it's associated with a clay mineral or not, whether it's plant-sized or very small size. So this particular organic matter versus mineral-associated organic matter is one way of fractionating soils. So when I say particulate leafy residues, that's like what you see, right? These semi-bits and roots and fragments, that's particulate organic matter. As it's broken down, you get material that looks like there's nothing in there, right? This looks like sand and clay. There's more carbon and nitrogen in here than there actually is in those fractions. You just don't see it. You can separate based on density. You mix soil with a dense liquid, and what floats is the light fraction, more leaf residue type material. The dense material is the more decomposed, likely mineral associated stuff. You can also separate at a higher size fraction, like the aggregates, whether they're in large or small aggregates. I can do all of these methods, and there are benefits and trade-offs, but what I generally am using these days is this particle size-based separation, where we separate based on its association with sand, silt, or clay. We get the sand size material as particulate organic matter, and then you get the silt and clay size stuff that is mineral associated organic matter, with sort of a spectrum of the extent of decomposition. You also have a spectrum of chemical complexity and carbon to nitrogen ratio, where here you have more carbon-rich carbohydrates and lignins in the leafy material, right, that's higher in carbon. And then as you get smaller in size, as it's decomposed, you get more of the nitrogen-rich microbial residues that bring that C to N carbon to nitrogen ratio down, okay? And so these are sort of measurable differences, but they're also associated with different functions in ecosystems, where particulate organic matter is considered this more bioavailable, labile source of nutrients. It's a nitrogen carbon source, and mineral associated organic matter has this slow turnover time. It's considered largely unavailable to crops. It's physically and chemically protected due to those mineral organic associations. And so there's a lot of interest, and in some of my work as well, we're trying to understand sort of the distribution of carbon and nitrogen across these different pools. When we, here just look at the amount of nitrogen out of total nitrogen, how much nitrogen is stored as particulate or mineral associated organic matter in the high and low soil organic matter soil. You see over 70% is usually in this mineral associated fraction, much smaller than this particulate fraction. We see the same across different biomes, that this is mineral associated organic matter carbon out of total soil organic carbon. And there's wide variation based on ecosystem, but in general, over half of the carbon is stored in this fraction. So really important reservoir that we also, slow to advance, want to manage for increasing the carbon and nitrogen storage capacity. So this is some work where here on the left, we're looking at, and I won't go into great detail, but it's just showing how there's a lot of interest in just looking at how, say, cover crop or different types of tillage, this is chisel and ridge tillage, how they impact then the nitrogen or carbon that's stored in those systems. And so here we saw that even just 5 years of, in this case, cover crop and a more conservation tillage approach, you see an increase in this mineral associated organic matter fraction. Here we were looking at 50 years of nitrogen fertilization and looking at more that long-term legacy of fertilization, how it impacts the storage of, in this case, nitrogen in either fraction. So the question of how to build this pool and form it more efficiently is work that I hope, it will continue in. But honestly, what I'm most excited about is this part of the story, which is that this mineral associated organic matter pool that's very slow and persistent and has on average very slow cycling turnover rate also contains this fast cycling, sometimes we call active fractions, where we know some of my work and other work has shown plant root microbe interactions can actually mobilize some of this mineral associated stuff in the rhizosphere, which I might use that term, I'll try to avoid it, but in case I say it, it's this zone of soil immediately surrounding roots under the influence of roots, where you have root exudates, simple carbon compounds like glucose and oxalic acid that all plants are releasing. You don't see it. There's the roots, but then the roots themselves are releasing these compounds in solution that are, there's a wide variety of this whole cocktail of stuff that they're releasing, but it includes often glucose, organic acids, and these can mobilize and destabilize mineral-associated organic matter. So I'm very excited by sort of understanding this more dynamic property of mineral-associated organic matter. Just a bit more about that plant mediated mobilization. Like I said, plants are known to release these exudates, and they can be microbially mediated or sort of directly destabilizing the chemical bonds. And we now are realizing that mineral organic associations are actually quite sensitive to these chemical biological changes that are happening in the rhizosphere. And it's something we have to account for and something that I'm also continuing to look into. There's also more and more research showing that mineral associated organic matter is quite sensitive to climatic shifts, especially in soil moisture, where climate variability or moisture variability can alter the stability of formation and stability of mineral associated organic matter and potentially increase its vulnerability to these plant mediated, rhizosphere exudate mediated pathways that I mentioned. And so this sort of climate sensitivity, vulnerability side of the research is somewhat new, and I want to continue to work on here, but it's really motivated by this problem, which is that a lot of systems here in the southern Great Plains, where Oklahoma is, all around the world, there are certain areas where we're seeing longer periods of drought, and then we have sort of sprinkled in these intense, extreme precipitation events that can lead to multiple days of flooding. And I just, a lot of us are trying to understand how organic matter is going to respond. Could there be potential shifts in the persistence, the stability, the bioavailability of the carbon within it and the nitrogen within it? And that is in this sort of understanding that soil water is a major control on organic matter formation and turnover. You need water to drive biological microbial activity. It's facilitating interactions between microbes, enzymes, and their substrates. You also need water to bind organic compounds to mineral surfaces. So it's really, this very important solvent in ecosystems, but especially in terms of the formation of mineral organic associations. And I think this shows that very well. And I'll start here on the right. This is looking at carbon content of different soils around the world in different biomes. And this you can think of as soil moisture increasing to the right. And we don't really see a very clear, or strong correlation between soil moisture and just the total carbon content in soils. But when you look at soil moisture and the carbon that's associated with iron and aluminum bearing minerals, these are those reactive clay-sized minerals that I was talking about. You see this very clear and strong association between the amount of those iron and aluminum bearing minerals and soil moisture, where if you're in this range, especially here, you can imagine a shift in soil moisture patterns might have a very strong impact then on how much carbon is stored in these otherwise stable, strong iron and aluminum bearing association or iron aluminum associations. And so I've been thinking more about this and understanding the consequences of moisture fluctuations for both how organic matter is formed on these mineral surfaces, but then it's vulnerability, sensitivity. The carbon that's entering soil and forming and becoming soil organic matter today, moving forward, might be very different from the carbon that's, a product of the last thousands of years in soil. And I think we have to think about it differently, where I think it's possible the mineral associated organic matter that's formed under these sort of fluctuating moisture environments may become more vulnerable to, say, the mobilization by plant roots and microbes or other sort of pathways that we're starting to explore. And so with the last few, 5 to 10 minutes, I just wanted to go through an example of sort of the type of experiment I've run and will continue to run, so you can start to imagine the experiments that maybe you all might find a way to tap into or find interest in. And I'm often working at the scale of a microcosm or mesocosm, so lab, bench scale, or maybe in a larger incubation chamber, some version of soils in a jar or some vessel closed so I can measure gas that's being released. And then these jars are destructively sampled over time for a whole suite of different soil microbial chemical properties. But in this case, this experiment is showing that in the jar I have mineral associated organic matter mixed with sand. So here I'm isolating that mineral associated organic matter fraction. I'm adding to it sand, carbon-free, sterile sand to make it more, aerobic and hospitable for microbes. And this mineral associated organic matter is isolated using this fractionation technique that I'll have set up in the lab, among other fractionation techniques, to isolate these particle size fractions of organic matter. But so here in this system, the only source of carbon and nitrogen is the mineral associated organic matter. It is, fairly highly disturbed, somewhat artificial, right? It's not an intact soil. There's trade-offs to this, but this is sort of a first step in the mechanistic work, right? To simplify the system. So that's the sort of experimental unit is this jar. And what here I've done is taken 2 sets of these jars. I took these soils and added to it an isotopically labeled amino acid. So this has carbon 13 label, this heavy carbon that has 1 extra neutron, making it very slightly heavier than carbon 12. That's a more abundant form of carbon, but stable isotopes allow us to, you can distinguish that mass difference and take advantage of it and use it as a tracer. So you can follow this carbon through the environment. If you use a tracer, you can then follow it into the microbes, into the mineral surface, into the atmosphere, CO2. I love using stable isotopes. I'm also showing that because I think there's a lot of cool ways to transfer it over to a lot of the work that you all are doing. I also will have an instrument for measuring carbon isotopes in gas. Anyway, what I've done here is added this isotopically labeled glycine to soils and then imposed 2 different moisture treatments. One is this wet dry cycling treatment, and the other is kept at constant moisture for 3 weeks. And so this is mimicking or trying to simulate these stream fluctuations in moisture. Obviously highly simplified and maybe not realistic, but this is the first sort of step towards answering these questions, I think. And so following that wet dry cycle, so I'll go back real quick. While these soils are incubating, they receive glycine here, and then the microbes are using it. It's sort of absorbing to mineral surfaces, desorbing. There's a lot of this cycling going on throughout these 3 weeks. That amino acid is moving through the microbial biomass and becoming now part of mineral associated organic matter. So you have at the end of this incubation, then some mixture of the original mineral associated organic matter and this new glycine derived organic matter. So anything that's moving through the microbes, anything that directly sticks to clay surfaces, that's now technically newly formed mineral associated organic matter, and it's isotopically labeled, so I can distinguish between the new stuff and the old mineral associated organic matter. Okay, and so the question here, one of the questions we wanted to answer is this mineral associated organic matter that's formed under those wet dry cycles, is it more vulnerable in general to decomposition, but also to plant root induced mobilization, this for exudate mediated pathway that I've talked about. And so this biological stability test we sometimes refer to, we add simulated exodus. We add glucose, oxalic acid, the two in combination, of water as a control. And then we measured a bunch of things, but I'm just showing one figure to sort of highlight one set of results. We measured the total CO2 that's respired after adding these, and also its isotopic composition. So that, by measuring its isotopic composition, we can say out of the CO2 that's respired, how much is from the original old mineral-associated organic matter, and how much of that CO2 is from the new mineral-associated organic matter. Okay. And so, just highlighting one set of results with the CO2, we're looking at mineral-associated organic matter carbon respiration, and it's separated by new mineral-associated organic matter respired and the old original mineral-associated organic matter respired. These brown sandboxes are the wet-dry treatment. The blue ones are the constant moisture treatment. And then they're separated by the control, glucose, oxalic acid, and the two in combination. And so, in general, what we found is that both for the new and the old mineral-associated organic matter, wet-dry treatments, especially in this Wisconsin soil, wet-dry treatments increase the respiration and decomposition. So there's a clear effect of, this moisture variability may be, increasing its sensitivity and vulnerability, both new and old. But we also found that the old mineral-associated organic matter was more sensitive to these simulated exodus. It became more susceptible to decomposition, which is now leading me to, some follow-up experiments and work to understand how prolonged exposure to these wet-dry cycles and exodus could potentially lead to a net loss over time in mineral-associated organic matter. This is only over 3 weeks in only 2 soil types. It's highly controlled, mechanistic, but there's ways that we can expand across multiple soil types, potentially do more in situ, in place work where we can manipulate at a field level or more mesocosm level the moisture dynamics, so they're more representative of true moisture changes. But this at least I hope gives you an idea of the types of experiments and measurements that I use. And so I'm also thinking if these altered moisture patterns are going to then compromise some of these short and long-term gains in mineral-associated organic matter that we're striving for, whether it's for increasing carbon capture and keeping it in agricultural soils, for carbon sequestration and climate change mitigation. That's all great if we're monitoring increases in carbon, but then if we're having also these extreme changes in climate, that could lead to very, very sudden drops and that sort of offset or now undermine all that accumulation that was monitored or that happened due to different management systems. So this sort of balance between accumulation and decay is what I'm really honestly most excited about. And so some concluding thoughts. So research has focused on building soil organic matter. It's a good thing. It's to have more. You're increasing carbon sequestration. You're improving the physical qualities that help mitigate different types of disturbances. And so there's often this emphasis on these 2 different pools having 2 distinct functions. Mineral-associated organic matter is a slow cycling, more inert pool. It's less sensitive to management. And so in theory, more carbon in this protected pool is a good thing, but we also need the decomposition of soil organic matter to deliver nutrients to plants. And so this sort of, contradiction between carbon stabilization and decomposition and nutrient delivery is something that we still don't fully understand how to optimize that. And so this division is helpful in terms of communicating and understanding organic matter, but I think it's also led to particulate organic matter being considered as this, primary nitrogen source. It's more bioavailable and cycling fast, and then we have mineral-associated organic matter and carbon sinks. They should be binned that way. But what I'm excited to think about is how we balance both carbon storage and nitrogen supply in soils, and I think we need to harness the multi-pool nature of mineral-associated organic matter, because we know that some of it is yes, very persistent and stable, but there's also this active fraction. Plants can access, microbes have a way to get at it. So I think that's, what I will be thinking about a lot in pursuing and build my program here. Among other areas of research, I just wanted today to highlight this soil organic matter centric area of research. So with that, I will just acknowledge folks that have directly supported, helped generate data at OSU, or where I was an assistant professor previously for 3 years, Oklahoma State, OSU, which is there's a few OSUs, and then at the University of New Hampshire, where I did my dissertation work, and many different undergrads and fellow grad students who helped with that work, and multiple funding sources as well. So thank you, everyone, and really happy to take questions. Yes.
I have to present genetic modified organism class today for agricultural lecture 321. Do GMOs have an impact potentially on productivity or composition, and is that an area of interest or study that is natural for?
Yes, I mean, it depends what they've been modified for, but there's certainly ways that they're genetically modified specifically to, deter pests, right, or resist certain, weed control. That, those sorts of elements of modification are meant directly to support productivity. But I'm not sure actually on modifications specifically. I bet they exist for supporting more this, organic matter production side. I'm curious about that. Where the line is between just regular crop breeding and modification is also sometimes blurry. Yeah, but no, it's an interesting area. I need to learn more about it. Okay.
In those biological stability experiments, is that what you call them?
Mm-hmm.
You talked about the MAOM being decomposed, the microbes in the biology. Where are the microbes in that system coming from? Are you taking sterile sand and then they're just coming through with the organic matter as that's separated?
They're just resident. They went on for the ride in the fractionation, and they're the ones that survived all that sonicating and centrifuging and oven drying and grinding, and so there's that issue of who's actually remaining in that mineral-associated organic.
Yeah, that's part of my question, or what I was kind of trying to get at. And then also is that it seems like the whole system is focused on the rhizosphere, right? So are you trying to isolate more of that rhizosphere soil for that, or is it bulk soil, whatever you get, just to give as much information as you can?
It's both. I guess I'm stimulating the rhizosphere. So in that soil, 20-30 grams of soil, I'm adding 200 microliters of a glucose solution, and so the whole soil sort of represents, it's not entirely accurate and representative, but that whole soil is supposed to represent rhizosphere soil versus the control water only is the bulk soil. But we could collect soil in the field from that adhered to the roots, that's rhizosphere soil, and that is like a foot away from a plant would be bulk soil. So that's something, there's trade-offs to go.
Yeah, that's what I was curious.
No, it's all, these are all really good questions. So the question of inoculating or not, or how to manipulate the community, we could add a whole bunch of layers to it and
that rhizosphere is going to be different on different roots too. So.
Oh yeah. It's messy.
I just recently got thrown in a new project, and I'm trying to figure out pretty much how your research relates to what we're doing. Pretty much what we're being tested or challenged to do to go onto the environment and find out these blue carbon ecosystems that sequester this carbon. One of the questions I had for you, what are some key components, where there's like some measuring or some monitoring of one, should you bring it to them into the field to look at these environments that already have the capacity to sequester more carbon?
And what kind of ecosystems will you be
Well,
looking at?
like also on the coast.
Right, so very similar to the types of measurements you mentioned, but the sediment in those systems and the water itself track measuring total carbon and then the composition of that carbon. So there's, I think, some work in the blue carbon world is similar to this, like the stability of the carbon within it. There might be differences in the methodology for wetland soils versus upland soils, but, so some probably things you can do with that, the DOC, the carbon that's in the water itself, carbon that's in the sediment, carbon that's expired, gas, those are the main tools that are. Yeah. But we should talk some more. Yeah.
Yeah, I was very interested in the wet, dry cycle
Mhm.
stuff you're doing. I was watching Jim Cantori this morning on the Weather Channel, and he was showing areas in Mississippi and Louisiana have now gone 203 days without a 1-inch rainfall, and how dry it has become.
Right.
So is there a lot of knowledge with this changing climate of where we need to be concerned? I mean, I know we have the moisture, the drought monitor and all that, but has anybody tried to relate the drought monitor to these major changes in cycling within the soils themselves? That seems like would be a pretty interesting thing to try to correlate, because it would really help farmers better know when to, take bold action, I guess.
There's definitely work in this area. I have one slide I didn't show, but just real quick, as an example, these are different systems with varying crop rotation diversity, so increasing diversity going to the right, and they were measuring yield drought resistance. So there was increasing drought resistance with increasing crop rotational diversity in this case, and they were able to link it to there's a moisture, the soil organic matter affecting yield indirectly through this plant water status. So yes, there's people sort of in specific systems or regions or in certain contexts working on this. I think it would be really neat. I think I've talked to a couple people about this, is try to identify certain soil types that are more vulnerable to carbon loss, and that might inform how to manage it, how to, different types of land use change. If you're switching from sort of cropland to forest or agroforestry, there's some systems that might become more or less prone to carbon loss under the changing climate. So having some sort of almost mapping or monitoring based system that can tell us that.
Using the soil curve number is what we use to develop urban runoff models, by using curve numbers and how different land, like imperviousness, how it would affect the soil. So you come up with a modified curve number. And so I'm thinking something like that, since you already have curve numbers for all your soil types, that would be a way to, maybe think about linking it, because we did something similar looking at urban runoffs.
Yeah, we would need to run that sort of test across the wide, so have a database of different soil types to see how they're responding to these moisture changes. That would be really, would require some modeling. So yeah, for sure.
Following up on Dr. Scott's questions, when I think of soil, like when you grow corn or something, it seems like it's often very dry. So microbes, being very important, undergo transient desiccation. Do people measure the water activity in the soil to see if, even though it's dry, there is still water sustaining the microbes and their biomolecules?
Some interesting work on that, especially with sort of biofilm production and that thin layer of water that persists even in very, very dry soils. If you go down just an inch, there's some moisture there, and the microbes are in, they're all concentrated through all the solutes and all their byproducts and the biofilm that all their osmolytes are starting to pump out. That's something we should chat at some point about ways to measure that in a useful way.
So they're inherently adapted to wet, dry.
Well, some more than others. I think that, depending on their physiology, and there's different species are more and less drought tolerant and have different byproducts that promote that. So, it's an interesting area.
I haven't maybe simulated like the rise of rhizosphere based on the roots and stuff. And I remember seeing someplace that the depth of the roots can really influence water absorption and nutrient circulation, things like that. So with your work, I'm presuming you're just using standard the measurements you're putting up there, but you're using it like the standard default settings. Do you plan on like modifying those to see does the amount of carbon change depending on the root depth?
Yeah, no, that sounds really interesting. Because I was taking soil just from the surface. I think it was, 10 centimeters or so. And then, there's the carbon and the mineral surfaces there, and the surface 10 centimeters are very different from what's happening, 50 to 70 centimeters. Microbial communities are different, the types of clay, relative amounts of those mineral particles. And yeah, there are people showing that depending on where you are along profile, there might be more or less carbon sensitivity to that rhizosphere effect that is in many cases mediated by the type of clay that's present. And
different layers.
yeah, so so there's that one picture somewhere that shows those layers. But yeah, if you haven't had a chance to crawl into different soil pits around, maybe I can take everyone on that. We should, Gabe and Buzz, dig some soil pits around and we can show people. But usually what you'll see, depending on the type of soil and the extent of weathering, the age of the soil, the climate, there's all sorts of things, but usually you see some type of horizonation. It's referred to where you have layers of soil that differ not only in organic matter content, so this is darker because it has more organic matter, but it's also differences in the mineral, the sandsilt clay composition. You have decreasing carbon as you go down, differences within these layers, that, an aggregate structure and all sorts of things. And that's what goes into like the soil taxonomy is separating soils based on these characteristics, how deep the horizons are, their color, their, if you add acid to it, does it fizz, are there carbonates present, that is part of like a diagnostic test you do to differentiate different soils in these layers.
This has been fantastic. Thanks so much. So as a nutritionist, I'm always thinking about, the nutrition of soil and and you mentioned it towards the end, this kind of competition between carbon sequestration and whether or not it goes down or right. I'd love to probably just chat more about that with you later. What that entails. It's just something that's presented, at least to me, in this context.
Yeah, especially the nutritional value of the food crop itself, I think, is overlooked. There's a lot of scratchy people on Twitter who will say there's no link between soil health and food quality or nutritional value, but I think it's just understudied.
Yeah.
So it's
Yeah. Another kind of behavioral health part you mentioned. So totally understand, if water is not able to seep down into the soil, it's just going to run off. Is there any evidence, or has anyone brought up the idea of the contribution of soil composition and perhaps wildfire risk?
Oh, I'm just meaning like that's a cool question, but so
I'm just thinking about like the wildfire, how that may relate to our world and climate resiliency. Is there any?
Definitely connection. I know there's people working on it. I can't like see it in my brain exactly who's doing it and what they found, but I know, there has to be a connection because soil organic matter is influencing the moisture retention, the hydrophobic qualities of soil as well. And so I can follow up. I'm actually the sort of wildfire part to this is also an area I'd like to go into, but it's totally new for me. So So it's an interesting area. We'll talk all these things. Yeah.
I was gonna say, this morning, y'all may have heard about all the terrible accidents in New Orleans recently on the interstate. They're coming from fires, and those fires are burning in soils with humid and very wet. And I did not realize that all the fires in Canada are also in wet environments, which is kind of why it's been hard to put them out, because they're burning underground. And the fire chief in New Orleans, they are pumping 1 million gallons of water an hour on that fire. That much water they are trying to pump to put that fire out. So that's a really good question about the connection on vulnerability of soil. Because if it's underground, it's much, these Picosin bonds are usually where the fires burn underground, and those are very difficult to put out. So,
Yeah, what about the soils contributing to risk or helping mitigate?
Yeah.
Interesting to think about.
I had a comment on that. It's good that you've got this slide up. What we're finding on prairie soils is that fire and grazing livestock, especially ruminants, is an increasingly important tool that we can use to increase the amount of pore space. So basically create soil aggregation. That soil aggregation then facilitates infiltration. So I find that people outside of the soils area or the ag area don't know much about it. And often people's instinct is immediately to go ahead and till. And immediately as we till soils, it's essentially a catastrophic event for microbes. So that's just a comment. And, management does affect the ability of infiltration. And to your point, Jed, I've been able to measure infiltration on the same soil series, and one, you might not be able to get an inch to go down in an hour, and then you go on the same soil series that's been completely differently managed, and you can't get the stopwatch going quickly. It infiltrates in 7 seconds. So soil is a dynamic system, and then even, you can take an infiltration rate 4 foot away, and it might be very different to us.
Yeah, not to complain about firefighters, but the first thing they do is they plow a fire line.
Well,
I think you got to do that.
Yeah, that's important.
Yeah. But on the other hand, you need to be thinking about what you just said about disturbing the soil stuff.
Management changes soil. I'm sorry.
No, don't
Management changes soil properties, and I don't think we appreciate how much that happens. My 2 cents. I had one other question, or 2 other questions.
Yeah.
If you've got a mesocosm, I think it's essentially a closed system. How do you vary the moisture content?
Well, it's not always closed. We opened it up and check the moisture and monitor it. But so if we need to manipulate something that requires opening it, then when we're measuring gas, it's during these windows of time, and then we sort of interpolate between and fill the gaps that way. There's always different parts of the system that we have to make compromises
Right.
based on.
Just for me, because I've never seen anything like that. Can you kind of physically describe what a mesocosm looks like? Is it like a jar? You kind of
Usually mason jar,
mason jar.
pint size, or whatever the smaller 240 mils is. And with the lid that it comes with, and then we hit, with a hole punch, those metal hole punch things, a hole out of the lid, and then put a rubber septum in there, seal it in place, and that's where a syringe with a needle is used to pull samples. But that's one size. I also want to do in the controlled environmental chambers, larger scale, more true mesocosms, which a micro mesocosm division, it's kind of blurry, doesn't matter. But the larger scale is like a PVC pipe this big, maybe this tall, where I have more room to manipulate sort of the depth, those depth, or maybe a plant can be grown in it. I can't do that in a jar. So that's still mesocosm, but I usually get fewer experimental units available with that. In the lab, in a mason jar, I'm anywhere from 100 to 300 jars. In a mesocosm with the PVC pipes, it's probably no more than 40 or something like that. Just to help you visualize. Yes, Laura.
I'm hearing more and more about water reuses and possible water reuses being used essentially for human drinking water, but also for agriculture and things like that. Is that something that you're going to like incorporate in? Because obviously those water sources are going to have various essentially microbes in them themselves are then being added to those soils, which you would presume they augment the microbial community as well.
Yeah, the irrigation water reuse is, I'm actually wondering here, is that common, like on irrigated soils or?
We're just taking rainfall water and putting it back.
Or even just through wastewater treatment being put into.
I know nothing about that one, I know it's text.
They do that on golf courses, they apply secondary treatment.
And some parks now as well. And I think there's discussion about some agriculture areas because the water becomes so low. They figure out or to continue agricultural purposes.
Right, I mean, you make, very important question of what microbes is it introducing and how that might shift the function of the soil. For agricultural irrigation water reuse, I think the main concern is chemistry of the water. It can start to be imbalanced, have high salts and, other, just becomes an issue sometimes, especially in more arid systems. But that's interesting. I don't know what to think of that, the sort of wastewater reuse. I don't know how do they process the water before it's
I couldn't say one person certainty. I know that it depends on where you're looking, but sometimes they can have like wetlands that goes through, and then somehow they're taking it that and bringing it to their places. I don't really know the American system very well, but it was something that was like they were saying how in Texas the water use varies so much across the state and what's available, and bringing new systems online over the next like 10, 15 years, the populations are supposed to grow. Basically, the reuse of water is like really being investigated in places like the Czech Republic as well. Yeah, central. So I'm just wondering.
That sort of water use efficiency question is really important. I'm on a project where it's based in the southern Great Plains of Texas and Oklahoma that's brought soils here and I'll continue working on, but part of that project is looking at how grazing and cover cropping and cotton systems might help improve water use efficiency, so we're not having to rely on sort of irrigation recycling, or have to deal with those problems, I guess, much. Because it's best.
Those are examples that are not always taken out.
Yeah, I know, that we're monitoring for. Another related to that area that I'm going into is biosolid application of lands and the microplastics and potentially other contaminants that it's introducing in soils, which is related to the wastewater treatment question. But I wonder how pervasive biosolid land application is compared to this other sort of wastewater that's treated.
Something like that.
Yeah, no, it's
I know human lives are like they were having projects and then the fact that they were growing cabbages. I just remember that much. It's stuck in my brain, and it didn't work the way they wanted it to work. Too much of the percentages that they were spreading it on. But it's interesting to think about all the ways to reduce how to use it effectively.
Yeah, Absolutely.
All the way around the car. I believe next week we're back. We have, video shields from DPEC, presenting on the environmental public health practices data platform. They have the. Be there. Or here.
Thanks, everyone.
Figure that out, see if you're